Hi, everyone. Welcome to the webinar hosted by Mini4Sigma. My name is Cindy Chen. I'm a scientist in an R&D group that has been designing, creating, and launching various microfluidic cell culture devices and plates for live cell imaging. Today, we're going to talk about the CellASIC ONIX2 Microfluidic platform and how we use it to capture real-time, high-quality images on growing yeast and bacterial colonies during the experiments. And here's just an overview of what we're going to cover today. First, we're going to talk about why it is important to do live cell imaging. Then I'll introduce the microfluidic platform and how we use it. Lastly, we're going to talk about the applications and what types of experiments you can perform using the system. So why do we want to do live cell imaging? There are tons of endpoint assays available in the market these days, and they surely give us some important data to understand how cells work. But more and more researchers don't find them sufficient enough to draw any conclusions because they don't see the full picture. For example, there's a cell right there at time 0 and still there 24 hours later. Does it mean the cells didn't move at all, or maybe it moved for a while and then moved back? Just because you didn't see it move didn't mean it didn't move at all, correct? So how can we see everything that happens in one experiment? The answer is live cell imaging. So it allows us to answer questions like when, the temporal information. So what were cells doing before and after the certain time points? And how did cells respond minutes after the treatment? Is it a fast response or a slow response? When were proteins of interest expressed? And then it also helps us ask questions regarding the spatial information, like the where question. Where were proteins of interest expressed? Was the protein shuttling back and forth between nucleus and cytoplasm? Also, questions like how. How does the dynamic environment affect biology? Did all cells respond to the treatment in the same way? Did cells recover after the treatment? How and when did they recover? This is all important information to help us view the big picture, and simple live cell imaging can help us with that. So take autophagy in mammalian cells as an example. When we starve the cells, they will form these microvesicles called autophagosomes to recycle the waste and to rebuild proteins that will help them survive through difficult times. Like these video clips, on the left is the control group where cells are normal in normal medium. You wouldn't see the accumulation of the autophagosomes. And on the right is when cells are starving in PBS. You see more and more green dots appear at 40 minutes into the starvation. You see when and where they're formed. And by using live cell imaging, you're confident to say, yes, the cells respond to starvation, and also the autophagy kicks in within 1 minute of starvation treatment. And now we use hypoxia as the stress source this time to initiate autophagy in cells. Then you plot the fluorescence intensity over time in a chart and find different dynamics in the cellular responses. And based on the piece of the data, you find that at some point, if you stress the cells with autophagy inhibitor, like chloroquine in this case, and prevent cells from recycling and rebuilding, the cells would be so stressed that even normoxia couldn't bring the cells back to the normal state. These are all the findings endpoint assays cannot provide. Here's another example of live cell imaging experiments. So you grow mammalian cells on our device. In this case, it's the human colorectal adenocarcinoma HT29 cell line. And then you introduce the invasive E. coli to study when and where the invasion occurs. In this case, HT29 is stressed by the invasive bacteria within 6 hours, and that eventually rapture 15 minutes after the treatment. Without live cell imaging, you wouldn't know how fast the invasion would take place. So now we all agree live cell imaging is important, and it gives you both spatial and temporal information that endpoint assays don't provide. But how do we do live cell imaging? For cell cultures, you'll need a cell culture device to hold your cells. You also need temperature and gas control to keep cells healthy. For the images, you'll need a microscope. There are some products out there to do this, but they're huge and often expensive. They basically put a microscope in the incubators, but that's just say you don't care about the size and the price tag. There's still limitations. For example, for a giant incubator like this, it takes 6 to 8 hours to reach equilibrium. After it reaches equilibrium, you would have to put the cells into the incubators by opening and closing the incubator doors. Finger crossing that by opening and closing the door, the gas content would remain the same. And some live cell imaging systems allowed you to treat the cells during your experiments, but the tubing going in and out of the incubators is just too difficult to set up. So here comes our unique CellASIC ONIX2 live cell imaging system. It's pretty small, about the size of a shoebox. It's compact as well. It comes with a reagent control system, a manifold that ensures temperature and gas control in the cell culture chambers, and then a culture plate to hold your cells. You load your cells onto the plate, hook it up with the manifold, and then you use your computer to control the perfusion, gas, and temperature. And then you use your microscope software to set up the time-lapse imaging under the microscope. And then you click on Start on both the perfusion system and your computer, and then walk away. You come back the next day and get data, and it's simple as that. So when you set up the experiments, it's going to look like this. You have the cell culture plate, and then you have cells and solutions loaded to the designated wells. You hook it up with the manifold that connects to the system. Then you use the software to control the pressures on the cell inlet well. So when the pressure is on, the media or the cell solutions in that well is going to route through the microchannels and go into the cell culture chambers. This is how you load the cells or treat the cells. Once you're happy with the loading results, you can then set up the treatment time, the gas, and temperature on the ONIX software. Place the plate on the microscope and use your microscope software to set up capturing intervals, autofocusing, setting up beacons, etc. And then press Start on the ONIX system. And also press Start on the microscope capture button. And now the system is going to treat your cells, change gas content, or change temperature at certain time points, all by itself, while your microscope is taking pictures on the cells. The even more unique feature is that we offer a wide range of microfluidic plates for different applications. We have mammalian plates, which allow you to culture mammalian cells, and then you can float in bacteria, like the host pathogen experiments we previously talked about. We have yeast plates and bacteria plates that are designed specifically to hold these microbes in place over time. But no matter what plate type it is, they all have a standard 96 well plate frame. There are 4 units on each plate so that you can use one as a control and do 3 other experiments, do 3 other treatments as the experiment groups. There are 6 upstream reservoirs, and the bottom is cover slip glass that is compatible with most of the inverted microscopes. Let's focus on the yeast and bacteria plates we launched a couple years ago. The main feature of these plates is that there is an elastic PDMS layer on the top of the cell culture chambers. When you load the cells with high flow of media, it pushes the ceiling to the top. When loading is done, the ceiling drops to certain heights, but narrow enough to gently hold the cells in place. Then even there's perfusion media flow, the cells will still remain in one focal plane. There are 3-tier ceilings on Y4C, ranging from 3.5, 4.5 to 5 micron. Y4D ceilings are taller, ranging from 5, 6 to 7 micron for diploid cells. B4A ceiling heights ranging from 0.7, 0.9, 1.1, 1.2 micron. So no matter what size your microbes are, they'll be trapped under one of the tiered ceilings and be held in place for the entire time. Without this trapping mechanism, if you're just taking an image on cells cultured on agar pads or on glass slides, you simply cannot capture a shot that is in focus, let alone monitoring the expressed or labeled proteins in real time. And that is definitely an issue ONIX can fix. Let's take a look at how researchers use our Y4C plates. Here's a clip from one of our customers in Japan used Y4C plates to study mechanisms behind chromosome and plasmid DNA dynamics in the cell. They're also interested in monitoring how cells form their shapes. Here's another testimonial. Another researcher, Professor Springer at Harvard, used ONIX Y4C plates to stimulate cells with 2 treatments and see how cells decide what to do. Also, they use the system to heat shock the cells and see genes turning on and off during perturbation. Let's hear him explain in his own words.
We're interested in how cells respond to heterogeneous environments. So typically when we do things, we change media, but it has both input 1 and input 2 there. But we want to see times if we have like input 1 on one side of the cell and input 2 on the other side of the cell, how does it integrate that decision? And is it integrated differently than when the cell is faced with a mixture of those 2 inputs across the whole cell? On some level, nothing that we discovered that we probably couldn't have discovered a lot of hard work in another way, but it allowed us to really test some things out and do things like and cleanly answer some questions that would have been difficult otherwise. And the 2 things that we got that were probably most interesting that would have been extremely difficult otherwise was doing a heat perturbation of a cell where we were able to look at the expression level of individual genes on a single cell basis before the shock and how that correlates with growth rate or response to the shock. And in switching between different media conditions, switching between a certain glucose-galactose regime to another regime, and looking at the response of the cells and their growth based on the genomic character of the strain and also its current expression level. So those are things that it would have been extremely, extremely challenging for us to do in any other way, and we're able to get some pretty interesting, exciting results by using the CellASIC ONIX system for that.
Another good application example is this clip where our customer's Professor Garner at Harvard talks about their research. They use onyx bacterial plate to study how cells form different shapes under different stress sources. Hear him explain his research.
So what the CellASIC system lets us do is a couple things. First off, it lets us actually follow the shape of the bacteria throughout time. And so we can look at the same cells over and over a very long period. So it allows us a constant perfusion of media. The other thing that's really useful is that what it lets us do is rapidly insult the bacteria, that is, rapidly add antibiotics or environmental stresses, or even turn on and off individual genes and watch, for example, how bacteria can gain shape or lose shape. So, for example, we have cells now which grow without any shape. They're basically amorphous blobs, and we can turn on given genes to actually reinduce rod shape and watch these blobs spit out rod-shaped cells, and we're watching the enzymatic motions of these processes as the cells are reemerging. So basically what the CellASIC machine lets us do is very rapidly turn on and off conditions or insults to individual things while following singles.
Besides Y4C, Y4D, and B4A plates, we have launched another unique plate for the Yeast customers last year, Y04T. So besides the elastic sealing, we add 3 elastic wall features to form a trap to hold yeast cells for up to 12 hours and more. Let's look at the trapping mechanism pictures on the right. When the cells are loaded with high flow of media, the cells will get randomly trapped in one of the traps. This is specifically useful if you're studying aging and just want to focus on the mother cells, or if you just want to study how long they can keep generating daughter cells. It also becomes very useful if you want to study mother-daughter interactions. Let's see the real trapping and culture in action. In the video on the left, it shows how loading is done. You see individual yeast cells gathering around the chambers and get randomly trapped in individual traps. And in the photo shown on the right of the slide, you see the results of a colony grown from one single mother cell in our plate after 24 hours of culture. And the special feature is that we designed the chambers in a way that the flow can easily wash away the excess cells. So now the culture can go up to 72 hours without clogging the nutrient or the treatment flow. And here's our newest plate, B04F, launched early this year, that is specifically designed for biofilm studies. There are 200 micron by 200 micron pads in one unit. Because of the elastic pad design, the inner ring of the biofilms will always be in monolayer and in one focal plane, so that no matter how many millions of cells are around, you can still clearly see the proteins of interest get turned on or off by perturbation inside the biofilm. And because there are only 2 pads with significant spacing in between, you can study the communications between the 2 biofilms that are some distance apart. And we're confident to say that B04F is the one and only biofilm plate that allows you to image 2 biofilms at the same time. And it's the only live cell imaging system that enables you to monitor the communications between the 2 biofilms. Here's a clip to show bacillus growing into a biofilm on our plate. As I mentioned in the previous slide, the inner ring of the biofilms are held in one focal plane throughout the entire culture. And we also observe the size of the biofilms changes over time, which is also known as the periodic growth of the biofilm. The size changes of the biofilm, or the so-called periodic growth, was identified by several scientists. One of them was Professor Sol at UCSD. He was able to use our previous prototype to trap and grow biofilms and introduced environmental perturbation and observed that biofilms could grow and reorganize and grow again and reorganize again. If you mutate this gene to remove the protein, the size wouldn't change. He published a paper on Nature and linked this protein with biofilm periodic growth. He also published another Nature paper using our system to study the membrane potential changes in the biofilms upon perturbation. During the making of this biofilm plate, Dr. Sol and his team have helped us identify the must-have features. They also helped validate the function of this plate, and now this plate is commercially available. There are many applications you can do on our plate, like protein translocation, protein trafficking. You can track certain proteins moving in the cell walls. You can heat shock the cells or introduce drugs, stress, or gas change and see genes turning on and off. You can study mitosis and how cells divide. And we have many customers that have published on Nature, on Cell, and great journals. And we hope your papers will be on our list in the near future as well. So this is it for this webinar. If you're interested in a product, please reach out to our field specialist or go to our website at miniportsigma.com/CellASIC to request a demo. Thank you for your attention.